Thank you, Dr. Corker, for organizing this conference. It's really great to be here. Thanks for everyone for being here today. And as he mentioned, lunch is next on the agenda, so that's good news. So when we think about how motion impacts emotion, I think we typically think about therapeutic effects of exercise. And that is how exercise can help reduce symptoms of current stress-related mental health disorders such as depression, anxiety, and PTSD. What we don't often think about are the prophylactic effects of exercise. This is the ability of exercise to prevent or reduce the risk of developing future stress-related mental health disorders. Because exposure to stress is such a primary causal factor in these disorders, we often just refer to this prophylactic effect of exercise as stress resistance or the stress buffering effects of exercise. And it turns out that exercise-induced stress resistance is very powerful. So here's one example. This was a meta-analysis published in JAMA Psychiatry in 2022, and these authors found that achieving the recommended amount of weekly exercise, which we heard is 150 minutes of moderate intensity exercise per week, this amount of exercise reduces the risk for developing future depression by 25%. Another study found that almost any type of exercise reduces the risk for future mental health burden, which includes symptoms of depression, anxiety, and PTSD. So these are huge protective effects against future stress-related mental health disorders. Despite these impressive effects of exercise, we actually understand very little about exercise-induced stress resistance. So one goal of my research is to try to understand how it is that exercise protects organisms from the mental health effects of stress. And so today I'm going to talk about that, and I'm going to show you the approach that we use in the lab to study this question. And then I'll discuss some of the mechanisms that we've identified that we think are important for the protective effects of exercise. And I'll talk about the stress-resistant brain. I'll talk about the role of serotonin neurons in a brain region called the dorsal raphe nucleus. And so yes, serotonin is involved in exercise-induced stress resistance, but probably not in the way you expect. I'll then talk about enhanced stress coping and the rewarding effects of exercise. And we're noticing some really interesting sex differences in these mechanisms. So I'll be pointing these out along the way. So how do we study this question? I mentioned that we don't really understand a lot about exercise-induced stress resistance. And one of the reasons for this is it's very difficult to do human clinical trials on stress resistance because we can't ethically expose humans to the type of stress that causes depression and anxiety and PTSD. So we're limited to observational designs. We can, however, use more definitive interventional designs in rodents by exposing groups of rodents to either stress conditions or no stress conditions. We can also really easily manipulate the physical activity status of rodents by simply putting a running wheel in their cages. If you ever had a rodent as a kid, or if your kids have a rodent pet, you know that if you put a wheel in a rodent cage, the rodent is going to run on the wheel. And it's probably going to keep you up all night long because they run at night. If you've never seen this, this rat will demonstrate what wheel running looks like. And you can see that there's nothing forcing this rat to run, right? It's not being chased by a cat. It's not late to an appointment. It's doing this completely voluntarily. And there's a lot of evidence to suggest that rats find, and mice and hamsters, find wheel running rewarding and reinforcing. So behavioral economics studies have found that rodents will pay a very high price for access to these running wheels. And if you take these away, the rodent gets very stressed out and aggressive. wheel running has actually been criticized in the literature as not being a good representative model of human physical activity because rats and mice typically run a lot, and unfortunately humans don't. However, I will argue that voluntary wheel running is actually a great, valid translational model of physical activity because similar goal-directed and habitual motivational processes govern physical activity in both rodents and humans. So what do I mean by this? So human studies have shown that typically humans that start engaging in exercise programs do this in a goal-directed manner. So there's a specific goal that humans are trying to reach. This could be a fitness goal, maybe weight loss, maybe they're trying to improve at a sport. And then what we see over time is only those people that develop a habit of exercise are the ones that can stick with exercise over the long term, regardless of what their initial intentions were. So it's not willpower that keeps people exercising over a long time. It's effective development of an exercise habit. And this seems to be true in rats as well. This is a typical pattern of voluntary wheel running in male rats. And what we see is that the escalation phase of wheel running, when nightly wheel running increases over time, is actually goal-directed. And then this plateau phase is habitual. Of course, one of the reasons we think this is that regions of the brain that control goal-directed behavior, namely dorsal medial striatum, prefrontal cortical loops, so these brain regions communicate with each other, govern wheel running during the escalation phase, and a region of the brain important for habit, the dorsal lateral striatum, or DLS, governs wheel running during the plateau phase. Of course, rats and humans have different goals for exercising, right? Rats aren't exercising to look good in their bathing suits. But we think that the reinforcer or the goal in the case of rodents is dopamine release in a brain region called the Nucleus accumbens, which can encode the reward value or the reinforcing effects of that exercise. And I'll come back to this in a few minutes. So these are data in males. It turns out that female rats run a lot more than males. Look at that, 10 kilometers a night on average. And perhaps more importantly, female rats escalate faster than males and reach the plateau, the habitual phase of exercise, a lot earlier. And we've been able to show that this is because females are actually more likely than males to use this habit center in their brain to control exercise. And that's why they're more likely to shift into this habit physical activity earlier or more readily than males do. And one reason I'm pointing this out is because we think that sex differences in the neural control of physical activity could actually mediate sex-divergent mechanisms. So the fact that males and females are using different neural circuits to govern exercise could influence the mechanism by which exercise enables stress resistance. All right, so now that I've hopefully convinced you that wheel running is a valid translational model of exercise, we can look at our typical experimental design. So what we do is we house rats with a locked wheel, no wheel or a locked wheel, or we have them housed with a running wheel. We then expose them to a stressor, and then we look at behaviors that resemble anxiety and depression. The stressor we use most often in the lab is inescapable stress, and I'll abbreviate this IS. And so during IS, the rodents are confined to a tube and they receive a series of electric shocks to the tail. These shocks aren't painful, they actually don't activate central pain processing circuits, but they're kind of annoying. The rats would rather these shocks not be occurring. And they're inescapable and uncontrollable. And a reason we use this stressor is because it results in behaviors in these rats that closely resemble symptoms of human stress-related mental health disorders. Now, it's important to realize that it's not these shocks themselves that are producing these behavioral outcomes. It's the fact that these shocks are inescapable and uncontrollable. That's the critical component. So this is a psychological stressor. And what we've been able to show is that rats that have access to a running wheel before stress are completely protected from all of these behavioral outcomes. So none of these behavioral outcomes occur if rats have a running wheel in their cages. So if you do have a pet rodent, put a running wheel in its cage. I'm going to show you examples of the 3 behaviors we measure most often in the lab. So you can see here that sedentary rats exposed to inescapable shock have a reduction in the amount of time they spend interacting with a juvenile rat of the same sex. They also show enhanced fear learning in a typical fear conditioning paradigm. Since social avoidance and exaggerated fear are common symptoms of anxiety, these behaviors in rodents are thought to resemble anxiety. They're used as a model of anxiety. Additionally, exposure to inescapable shock will produce a deficit in the rat's ability to learn to escape from a simple escape task in a shuttle box. This stress-induced escape deficit is a common model of depression, and it's sometimes referred to as learned helplessness. You might have heard of learned helplessness. And so what we can see is that if rats have access to running wheels before stress, these behaviors do not occur. Okay, so hopefully you agree with me that voluntary wheel running is a valid translational model of physical activity, and it prevents the anxiety and depression-like effects of future inescapable stress. And then it's important to remember that sex differences in the motivational processes that govern wheel running could inform sex differences in mechanisms underlying stress resistance. So that's what we're going to get to now. So it's very much the case that the brains of physically active organisms are very different from the brains of sedentary organisms. This is a list of just some of the changes in the brain that we've seen after wheel running in rats, and there are many others. In addition to these changes in kind of specific molecular systems, there are large structural changes in brain regions after exercise. Dr. Rand talked about these. Now, any of these changes could potentially be important in mediating the protective effect of exercise against stress. But the trick is to really know which one of these is causal, right? So just because exercise changes something in the brain, that doesn't mean that that thing that's altered is actually important for stress resistance. I know this is obvious, but we often forget this. And I'm going to show you 2 interesting examples of this. So 2 well-known effects of exercise are to increase brain-derived neurotrophic factor, or BDNF, in the hippocampus, and to constrain the activity of the HPA axis during stress. Exercise, or wheel running in rats, not only increases BDNF in the hippocampus, it prevents the reduction in BDNF caused by inescapable shock. However, this protective effect of exercise is not important for the the ability of exercise to prevent the behavioral outcomes, because wheel running will still prevent those behavioral outcomes even if we experimentally reduce BDNF. So we don't need BDNF for wheel running to protect rats from stress. The behavioral outcomes of stress anyway. Similarly, we know that wheel running reduces the HPA response to mild stressors. However, if we expose rats to a severe stressor like inescapable tail shock, we see that in physically active and sedentary rats, there's an almost identical increase in circulating glucocorticoids in both male and female rats. So BDNF and the HPA axis do not seem to be important for exercise-induced stress resistance. So since exercise produces so many changes in the brain, rather than looking at the causal role of each one of these one by one, maybe a better approach is to try to understand what produces the behavioral outcomes of inescapable stress in the first place, and then see if running changes those known mechanisms. Now, I mentioned earlier that the behavioral consequences of our stressor, inescapable tail shock, are dependent on the inescapable nature of the shocks. Here we have a pair of rats who are receiving the identical number, duration, and intensity of shock. The difference is that the rat on the bottom has no control over the termination of the shock. The rat on the top learns to control the stress by turning a wheel or pressing a lever. These shocks are yoked between rats, so that ensures that they both receive the same shocks. And what we see is that only the rat receiving inescapable stress shows depression and anxiety-like behaviors. The rat that learns control over the stress shows none of these behavioral outcomes. So maybe we can look at the brains of these rats exposed to stress of different controllability to understand what are the most important changes in the brain that lead to these maladaptive behavioral consequences. And when we do that, one of the things we see is that inescapable stress and escapable stress both equally reduce brain-derived neurotrophic factor in the hippocampus. They also both equally increase the activity of the HPA axis and glucocorticoids circulating in the blood. So it's not really surprising that these factors weren't important for exercise-induced stress resistance, since they're not involved in the behavioral effects of inescapable stress in the first place. So I've talked a lot about what isn't important for exercise-induced stress resistance. Now we're going to talk about what is important. So when Steve Mayer was first characterizing this stressor controllability model, his lab found that in a brain region called the dorsal raphe nucleus, which is a midbrain structure that contains a lot of serotonin neurons that project throughout the brain, there's actually a huge increase in serotonin release within the DRN. And this is not happening in the brains of rats with control over the stressor. It's happening in the brains of rats who lack control over the stressor. So the rats that show depression and anxiety-like symptoms after stress have a huge increase in serotonin release in the DRN. And it turns out that this serotonin is very important for producing the behavioral outcomes. And the way that this works is that the other regions of the brain drive activity of serotonin neurons in the dorsal raphe nucleus to such an extent during this inescapable stress event that there's a lot of serotonin release in the DRN. This serotonin binds to a receptor that's located on serotonin neurons called the serotonin 1A inhibitory autoreceptor. Now, this receptor typically serves an important negative feedback regulatory role over the activity of these serotonin neurons. But what happens during inescapable shock is that there's so much serotonin release that these receptors desensitize, and they downregulate, and they do that through internalizing into the neuron. This is similar to the way we just heard about the way endocannabinoid receptors desensitize. This leaves serotonin neurons in a sensitized state so that they now respond to the mild stress of behavioral testing with exaggerated serotonin release in projection sites of the dorsal raphe. And we've shown that serotonin in the amygdala leads to social avoidance and exaggerated fear, and serotonin in the dorsal striatum interferes with shuttlebox escape behavior. So this initial hyperactivation and subsequent sensitization of serotonin neurons is both necessary and sufficient to produce these behavioral outcomes. Because of that, we wondered whether exercise prevents these behavioral outcomes by reducing the activity of serotonin neurons that we see during stress. And to test that hypothesis, we used a technique called microdialysis, which allows us to actually measure serotonin release in the DRN during stress. And what we found, so here's the sedentary rats. This is the typical increase in DRN serotonin release that occurs during shock, inescapable shock. This doesn't occur if the rats had a history of wheel running. It's significantly reduced. These are brand new data in females. We've also observed this same effect after 6 weeks of wheel running in male rats. So in addition to reducing that hyperactivation of serotonin neurons, we've observed that exercise prevents this sensitization of these neurons and reduces serotonin release in these projection sites during behavioral testing. So this is the primary mechanism by which exercise prevents these behavioral outcomes. Interestingly, serotonin constraint isn't a stress resistance mechanism unique to exercise. So both exercise, learning control over stress, administration of prophylactic ketamine, as well as the experience of social dominance, all enable stress protective effects by constraining that DRN serotonin response. Okay, so I showed you that exercise produces many brain adaptations, but not all of these are going to be causal in exercise-induced stress resistance. What we think is the most important primary mechanism is constraint over activation of DRN serotonin neurons that occurs during this uncontrollable stressful event. Okay, so when we ask, how does exercise enable stress resistance? What we're really asking then is how exercise constrains the serotonin response. And so we're gonna be considering first the role of enhanced stress coping. So there's a lot of talk in the literature about exercise enhancing stress coping. But what exactly does that mean? What are the neural correlates of stress coping? And what does this have to do with exercise-induced stress resistance? So one definition of stress coping is the ability to mobilize thoughts and behaviors to manage stressful situations. And a particularly effective coping strategy is one I just described to you, learning control over the stress. And just like exercise, a history of learning control over stress actually protects organisms from future uncontrollable stressful events. So it not only doesn't cause these behaviors in the first place, it protects rats from future uncontrollable stressful events. And it does that by constraining the dorsal raphe nucleus serotonin response to the stressor, just like exercise does. And the neural mechanism underlying this stress protective effects of behavioral control is actually pretty well understood. Work from Mike Barada's lab at University of Colorado Boulder has shown that rats use dorsal medial striatum prefrontal cortical loops to learn to terminate that shock when they're exposed to escapable stress. This results in plasticity in the prefrontal cortex, which allows neurons in the prefrontal cortex to be active during the later time of uncontrollable stress. And what that does is it actually inhibits serotonin neurons in the dorsal raphe. So this whole circuit, this is a very, very well characterized stress resistance circuit. The prefrontal cortex inhibits serotonin activity in the DRN. We know that rats use dorsal medial striatum prefrontal cortical loops to govern wheel running. I told you that a few minutes ago, at least in males, right? So maybe this enables similar prefrontal cortical plasticity, which causes stress resistance after exercise. To explore the role of the prefrontal cortex in exercise-induced stress resistance, we injected a retrograde tracer into the dorsal raphe nucleus. This tracer gets taken up by axon terminals of the prefrontal cortex. It travels back to the PFC, and it labels these neurons red. We can then quantify their activity by looking at a neural activation marker called CFOS. And what we see is if the rats had access to running wheels before stress, this increases or potentiates the activity of the prefrontal cortex to dorsal raphe nucleus circuit during the stressful event. So yes, exercise recruits this inhibitory prefrontal cortex pathway. We can also see potentiated activity of the prefrontal cortex when we look at general calcium activity within the PFC using a technique called fiber photometry. I won't get into the details, but you can see that there's a lot more calcium activity in the prefrontal cortex of rats with access to running wheels before the stressful event. So these data suggest that exercise is recruiting the prefrontal cortex to dorsal raphe nucleus circuit, and that could contribute to stress resistance. Now, these are in males. When we looked in females, we were very surprised to find that exercise had no effect at all on activity of the prefrontal cortex to dorsal raphe nucleus pathway, nor did it increase at all prefrontal cortex activity in general. So what is going on here? Why would exercise use a mechanism involving the prefrontal cortex in males to enable stress resistance, but not in females? So remember, males are using dorsal medial striatum prefrontal cortical loops to control exercise, whereas females have a preference of using habit circuits, the dorsal lateral striatum, to govern exercise. This could be preventing exercise from enabling the prefrontal cortex plasticity that's required to enable stress resistance. However, female rats are still protected from stress after exercise, right? So how do we explain that? So one explanation is that there's an alternative mechanism that's recruited by exercise in the brains of female rats that is enabling stress resistance in the absence of the prefrontal cortex, or independent from this prefrontal to DRN pathway. We'll get to what that is in a second, but first we actually want to test whether this prefrontal cortex to dorsal raphe nucleus pathway activity that we see is necessary for exercise-induced stress resistance. And one of the ways we tested this was by simply removing the prefrontal cortex. This is something we can do in rats that we can't do in humans, right? So we took out the prefrontal cortex of rats, allowed them to run, exposed them to stress, and what we were surprised to find is that these rats, these were all males, were still protected from the behavioral consequences of the inescapable shock, despite the lack of the prefrontal cortex. So now what is happening? All right, so there seems to be an alternative mechanism in the brains of males as well that's enabling stress resistance independently of the prefrontal cortex. This is kind of exciting. The prefrontal cortex receives a lot of attention in the stress resistance literature about its role in stress protective effects. So it's exciting that we're seeing stress resistance mechanisms that seem to be independent of this known pathway. Okay, so summary really fast. It looks like enhanced PFC-dependent stress coping could contribute to exercise-induced stress resistance in males, but other mechanisms are likely involved in both sexes, and these mechanisms probably become critical in females who don't recruit that PFC to DRN pathway after exercise. So what is this alternative mechanism, and is it similar between males and females? This brings us to the role of exercise reward. So deficits in reward processing, thought to be due to dysregulation in dopamine signaling, have long been associated with stress-related mental health disorders. We know that wheel running is rewarding and reinforcing to rats, and some of us might think that exercise is rewarding as well. So a long-standing assumption in the exercise literature has been that the rewarding properties of exercise involving brain dopamine must contribute to the beneficial effects of exercise on mental health. However, exactly how this works has not been shown yet. So Jerome Muehl, while he was doing his postdoc with Laurie Goodyear and Eric Nestler, came pretty close to explaining this when he found that molecular and synaptic plasticity in a brain region called the Nucleus accumbens, which is an important brain region involved in reward processing, is critical for the ability of exercise to enable protection against the behavioral effects of social defeat stress. This is another model, animal model of depression. So it does seem that a brain region involved in reward is important for exercise-induced stress resistance. But what was left unanswered is what the signal was that contributes, that leads from exercise to nucleus accumbens plasticity. Another unanswered question was the circuitry. So how is the nucleus accumbens plasticity actually enabling stress resistance? So these authors hypothesized the signal was dopamine, but whether dopamine is actually increased in the nucleus accumbens during exercise is unclear. So we set out to test that by using a technique called fast-scan cyclic voltammetry, which allows us to measure the oxidation of dopamine in real time as it's released in the nucleus accumbens during wheel running. And what we found is, yes, indeed, there's a huge increase in dopamine release in the nucleus accumbens during this wheel running activity. So one function of dopamine in the accumbens is thought to be encoding of reward value. So this increase in dopamine that we're seeing could reflect the reinforcing, rewarding effects of this wheel running activity. Okay, so the signal could be dopamine. What about the circuitry? How does the nucleus accumbens end up inhibiting the dorsal raphe nucleus? So there's actually a paper in Neuron from 2014 that characterized a direct neural connection between the nucleus accumbens and the dorsal raphe nucleus. This projection is inhibitory, and if you stimulate this pathway, you can actually inhibit serotonin neurons in the dorsal raphe. So just like the prefrontal cortex, the nucleus accumbens is anatomically positioned to be able to constrain serotonin responses in the DRN. So we asked the question, does exercise potentiate the activity of this pathway during stress, just like it does with the prefrontal cortex pathway? So again, we used the retrograde tracer, but this time we got to turn the neurons green instead of red. And what we saw was that just like the prefrontal cortex pathway to the DRN, prior exercise potentiates the activity of the nucleus accumbens to DRN pathway. Okay, so this pathway seems to be an alternative pathway that can enable stress resistance in the brains of males. That data was from males. And it could do so independent from the prefrontal cortex. So we next wanted to know if this activity was causal. Do we need this activity to enable stress resistance from exercise? We couldn't test that in males, right? Because they have multiple mechanisms. If we inhibit one, the other one compensates. We saw that with the prefrontal cortex. So what we did is we tested this in females, because as far as we know, females only have one stress resistance pathway that's recruited by exercise. So maybe we can inhibit this pathway in females, and that would restore the behavioral outcomes of stress. And so we did that, and the approach we used was called intersectional chemogenetics. So we used two viruses. One is injected in the dorsal raphe nucleus, one is injected into the nucleus accumbens. This is bilateral, both sides of the brain. And what this approach allows us to do is express a inhibitory designer receptor exclusively in neurons of the nucleus accumbens that project to the dorsal raphe nucleus. We can then inhibit these neurons by injecting a designer ligand called J60. So designer ligands and designer receptors theoretically don't do anything but communicate with each other. So this receptor is just sitting here waiting for this ligand. When the ligand is present, we can inhibit the activity of this pathway. And when we did that, what we saw, which is very cool, is that we actually restored stress-induced social avoidance and exaggerated fear in physically active females. So this nucleus accumbens to dorsal raphe nucleus pathway seems to be critical for the stress protective effects of exercise in females, because if we inhibit its activity, we restore the behavioral outcomes of inescapable stress. So exercise produces neuroplasticity in the nucleus accumbens. This could be through reward-related dopamine release in the nucleus accumbens. And a consequence of this dopamine signaling seems to be sensitization of nucleus accumbens to DRN pathway during stress, which could be constraining the dorsal raphe nucleus activity. We know that this activity of this pathway during stress is an important mechanism mediating stress resistance from exercise, because when we inhibit this pathway during stress, it restores stress-induced behaviors in females. Okay, in my last few minutes, I'm going to throw a little curveball regarding sex differences. So it seems that males have a greater number of stress resistance pathways that are recruited by exercise, right, to enable this stress protective effect than females do. However, female rats are more responsive than males to the stress protective effects of wheel running. So if we allow rats to run for 6 weeks, that results in equivalent stress protection in males and females. However, if we only allow the rats to run for 3 weeks, that results in full stress protective effects in female rats, and it has no effect on males. So for example, here we can clearly see that 3 weeks of wheel running fails to prevent stress-induced exaggerated fear in male rats, but does prevent stress-induced exaggerated fear in female rats. We see the same thing when we look at serotonin release in the dorsal raphe nucleus. These are the data I already showed you, but what I didn't mention is that this is only 3 weeks of wheel running. If we allow males 3 weeks of wheel running, that does not result in the same DRN constraint that we see in females. This is kind of exciting, I think, because as far as I know, exercise is the only stress-resistant mechanism that is able to work better potentially in females compared to males. So what is going on? Why is this happening? So we don't know, but our current hypothesis is that there's potentiated dopamine release in the nucleus accumbens during exercise in females that's accelerating that plasticity in the accumbens that's necessary for stress protective effects. Consistent with that hypothesis, it's well known that there's potentiated dopamine responses in females than males. This hasn't been shown with exercise yet, but with drugs of abuse, with electrical stimulation. We have not yet looked at dopamine release in the nucleus accumbens of females during exercise, but we have looked in the dorsal lateral striatum, which, if you remember, is that brain region that's important for habitual control of exercise and habits in general. And what we see is that there's a much bigger increase in dopamine release in the dorsal lateral striatum of female rats during wheel running compared to male rats. And this could be the driver of that more rapid transition to habitual wheel running that we see in females. But our hypothesis is that we'll see a similar effect when we look in the nucleus accumbens. Okay, I'm just going to conclude here. So we've identified neural circuits that seem to be important for exercise-induced stress resistance, but which circuits are used depends on sex. Males seem to use multiple stress resistance pathways, the prefrontal cortex to DRN circuit, as well as the nucleus accumbens to DRN circuit, whereas females seem to just use the nucleus accumbens to DRN circuit to inhibit the DRN after exercise. Females are more responsive than males to the stress protective effects of exercise, and this could be due to enhanced dopamine release in the nucleus accumbens, which would accelerate that plasticity in the accumbens. So I think understanding these mechanisms is important, and studying this in rats as well, because it lends credence to the idea that exercise reduces the risk of developing stress-related mental health disorders. So if we know how something works, then we're more likely to believe that, right? Also, rats are immune to the placebo effect. So rats aren't expecting the wheel running to protect them from stress, but yet it's happening anyway. And then also, I think understanding these mechanisms could inform approaches to optimize stress protective effects of exercise in humans. So if these data do translate to humans, and of course we don't know that right now, but potentially maximizing either the rewarding properties or the goal-directed control over exercise could most benefit men, whereas maximizing the rewarding properties of exercise specifically could most effectively work in women. So I'd just like to thank everyone who contributed to these studies, graduate students, postdocs, and our collaborators, Dr. Mike Baratta, who I mentioned, and Dr. Steve Mayer. So thanks so much. And I talked for a long time, so there's only a few minutes left. So thanks.